All right, ready to talk about spermatogenesis. I introduced this to you guys the other day. Fancy term that just simply just means production of spermatozoa, or fancy word for that is just, it's sperm, what we would commonly call it. That takes place in the seminiferous tubules, which are found inside the testes, and that's kind of the last little bits of what we talked about on Tuesday. Okay. This involves 2 rounds of meiosis, so 2 meiotic divisions, which takes place in order to reduce the chromosomes from 46 to 23, which we call the haploid number. And those sperm that would contain those 23 chromosomes would therefore be considered mature. This begins in a man's life at puberty, and I say man kind of loosely. Obviously, you could probably make a case for more of a boy at this point. But once they start going through puberty and they start this process of spermatogenesis, they are at that point where they could, impregnate a female. So really calling them man, I think, is pretty accurate. And this will continue throughout the entirety of the man's life, whether they're, 25, 30, 35, up into 105. Okay. It involves the following. Beginning with cells called spermatogonia, a type of stem cell located near the basement membrane of the seminiferous tubules, which are able to squeeze through the blood testis barrier, where they then go through developmental changes, forming into primary spermatocytes, which are still diploid in number. So we're still at this stage containing 46 chromosomes. If we jump to the next slide, we've got kind of 2 different pictures showing you. Here's where the basement membrane is that I just mentioned. Here's the spermatogonium, which is just the singular form of this word, spermatogonia, that was on the previous page. Those transform into primary spermatocytes. Okay, and we can see the same thing here. And remember, we use the abbreviation 2N to describe those cells that are diploid in number. Shortly after forming, those primary spermatocytes replicate their DNA and begin meiosis 1. And upon completion of that, 2 haploid secondary spermatocytes are formed. So we had 1 cell that was diploid. Now we've got 2 that are haploid, meaning that they now only have 23 chromosomes. And we see that in both pictures, left and right sides of the page, here on the next page. Okay. Those secondary spermatocytes then undergo the second round of meiosis, which results in the formation of 4 spermatids, and those are still haploid. So we don't reduce the chromosome number because that's where we separate the sister chromatids. Okay, now a unique process occurs during this spermatogenesis process in which the spermatids actually fail to complete cytokinesis, which leaves cytoplasmic bridges. And we can see that in these couple of images. Remember, cytokinesis is where the cytoplasm of a cell splits in order to basically break the 2 cells away from one another. Well, that doesn't actually take place in meiosis 1, nor does it occur in meiosis 2. Those little connections, as you can see, one is labeled right there, is called a cytoplasmic bridge, which may account for the synchronized production of sperm in any given area of the seminiferous tubule. So essentially, the thing is to make sure that all those sperm in the same area are going through the same stage at the same time. Okay. Final stage is known as spermiogenesis, not to be confused with spermatogenesis. Spermiogenesis is the development of those haploid spermatids into sperm, where essentially they go from these kind of more rounded cells with a tiny little flagella to the more mature sperm that you can see here at the bottom, where they've got that really long whip-like tail. The head is kind of in that cone shape, which is kind of a unique feature to those sperm cells. Notice that no longer are those cells connected to one another. Those little cytoplasmic bridges have broken free. Okay. No cell division occurs during spermatogenesis, except they just develop from the spermatids into the sperm cells. Okay. And then they're released from the Sertoli cells, involving a process known as, or event known as spermiation. So you got spermatogenesis, you got spermiogenesis, and you got spermiation. Those words are all fairly similar, plus knowing all the different cells and whatnot. And make sure you know the kind of order that things are going through, which cells are diploid, which cells are haploid. Know the difference between spermatogenesis, spermiogenesis, spermiation, and you'll be in good shape. Let's talk a little bit more about sperm, their kind of structural makeup and functions. Each day, about 300 million of these sperm cells complete that process of spermatogenesis, which is an incredible number, 300 million, considering it only takes one to fertilize an egg. So one sperm per baby that's produced, meaning that 300 million sperm every day is way more than necessary, because how often do guys impregnate females? Some never in their life, some a few times, some quite a few times, but not 300 million babies a day. Okay, so a lot of sperm are produced that just end up basically dying off. Size-wise, they're about 60 microns in length, containing several structures adapted for reaching and penetrating a secondary oocyte, which is the egg cell that we'll learn about here in a little bit. Each sperm consists of kind of two major regions, the head and the tail. The head is important because that's where you find the nucleus, which is where the 23 highly condensed chromosomes are. But it's also the place where you're going to find this structure called an acrosome, cap-like vesicle filled with enzymes that aid in penetrating the sperm cell into the secondary oocyte. So it helps with burrowing in, and involves a process that we'll talk about later in the lecture called capacitation, involving the functional changes sperm must undergo in the female reproductive tract, allowing them to fertilize that secondary oocyte. Then you got the tail, which includes kind of several different parts: a neck where it constricts, the middle piece where you find a lot of mitochondria giving it energy, principal piece, which is the longest part, and then a whip-like tail at the end. Basically, the tail is just built for locomotion, to be able to allow the sperm to swim. Thinking like if you've ever seen like videos of a snake swimming, or I'm sure you might have even seen, videos of sperm swimming when zoomed in under a microscope. They kind of have that pretty recognizable way that they move. So those are the sperm and the seminiferous tubules that we talked about the other day, where the sperm are produced, all of which was discussed inside of the testes, but aren't the only male sex organs. So let's talk about some other ones here. We have the epididymis, which adheres to the posterior side of the testis. And I would highlight this sentence because it's very important. This is the site of sperm maturation and storage. So this is where sperm mature and become functionally able to swim, and they will be stored. This takes about 2 weeks, where they acquire this motility and ability to fertilize an egg. So about 14 days. But sperm can remain viable here for up to several months. Okay. And any sperm that are stored here not ejaculated by that time are eventually phagocytized by cells in the epididymis. So chewed up and digested by probably macrophages that we've talked about before. Epididymis also contains smooth muscle fibers that will contract during ejaculation, forcing the mature sperm into what's called the vas or ductus deferens, which is a muscular tube that serves to deliver sperm to the seminal vesicles. This duct has a very narrow lumen as well, innervated by sympathetic fibers. And like the epididymis, it can store sperm for several months, but not to the degree that the epididymis can. Okay. Seminal vesicles are a pair of glands posterior to the base of the urinary bladder, and they secrete several substances into the seminal vesicle ducts, which will connect to what's called the ejaculatory ducts. These are formed from the union of the ducts from those seminal vesicles, but also the vas deferens, and it passes through the prostate gland, emptying into the prostatic urethra. And so that's its job: first force sperm into the urethra during ejaculation. Prostate gland is going to surround the urethra and ejaculatory ducts immediately inferior to the urinary bladder. Secretes a milky, slightly acidic fluid into the urethra, increasing sperm mobility and viability. And then the last glands of the male reproductive system I want to talk about are the bulbourethral or Cowper's glands, which are actually at the inner end of the penis, just below the prostate, where they can release a clear alkaline fluid that will lubricate the head of the penis and protect the sperm by neutralizing any residual urine. It's the acidity of that residual urine that would be harmful to the sperm. There's also going to be mucus in the fluid here that lubricates the end of the penis, which helps decrease the number of sperm that are damaged when a male ejaculates. This is where that pre-ejaculate fluid is coming from, and that's what comes, obviously before the powerful release of the semen, and that's why it's clear and not kind of that milky white color like the semen is. The urethra we talked about the other day because it is a shared tube of not just the reproductive, but the urinary system. So back when we talked about peeing and micturition, learned a little bit about it, but its job is to carry urine from the bladder during urination as well as semen during ejaculation. And then penis serves to deposit semen into the vagina. And during sexual activity, blood vessels in the penis will fill with blood, allowing it to become erect and able to enter the female reproductive tract. We'll get a little bit into more about erections and ejaculation here in just a second. Let's talk a little bit more about semen first, though. It can be described as a mixture of sperm and seminal fluid, typically having a volume of about 2.5 to 5 mils. And each of those mils would have about 50 to 150 million sperm. So, you take 50 million at the low end times 2.5, or if you're going at the high end, 150 million times 5, and that's going to tell you that every time a male ejaculates, lots and lots and lots and lots and lots of sperm are released, way more than necessary, obviously, to get a female pregnant, because it only takes one sperm to find its way into the egg to fertilize it. And it's going to be slightly basic or alkaline in pH of 7.2 to 7.7 because of the secretions from the seminal vesicles. So it's pretty close to our blood that we learned the other day as 7.4. Provides a transport medium, nutrients, and protection for the sperm. Will coagulate or clot after ejaculation due to fibrinogen proteins. The liquid portion of semen contains substances that help to nourish and activate sperm cells. If you want to kind of understand the ratio, it's about 10% sperm and about 90% other stuff. And that includes stuff coming from the prostate gland, which secretes about 1/3 of the total volume of semen in the urethra. And there's secretions, there will be vitamin C and several enzymes that help activate the sperm. Bulbal urethral glands produce mucus that neutralizes acidic urine in the urethra just prior to ejaculation, adds a little volume to semen. And then the seminal vesicles, which secrete several substances, which includes alkaline fluid, neutralizing acid of the vagina, fructose, which provides good energy to the sperm, prostaglandins that help stimulate smooth muscle contractions within the female reproductive tract, as well as to facilitate the movement of sperm cells up the tract, and then fibrinogen, which helps to coagulate or clot the semen after ejaculation, which makes sense if you remember, we've talked about fibrinogen before. That's that protein that we find in our blood that helps our blood clots form. So it's the same protein, just clotting a different type of fluid, obviously, not blood, but semen. All right, despite the old adage that men think with their penis and everything they do is relegated by the penis, okay, truly, men really do think with their brain, and everything that has to do with reproduction involves the brain, okay? The brain testicular axis refers to this relationship between the hypothalamus up in the brain, as well as the anterior pituitary hanging down from the hypothalamus, and then the testes. So literally it's the relationship between the brain and the testes. This is what controls spermatogenesis, which once again is the process of sperm cell production by the testes. At puberty, certain hypothalamic neurosecretory cells increase their secretion of gonadotropin releasing hormone. GnRH is its abbreviation, which is the hormone that stimulates the two gonadotropin hormones from the anterior pituitary. Those two gonadotropin hormones are luteinizing hormone and follicle stimulating hormone. And we've talked about those before, but here we're going to get into a little bit more detail about them. Luteinizing hormone is going to stimulate the interstitial cells within the testes to secrete testosterone. Testosterone is a steroid hormone, so it's synthesized from cholesterol in the testes. It is the principal androgen in men. Utilizing negative feedback mechanisms, testosterone suppresses secretion of LH and GnRH. So essentially, if testosterone levels are very elevated, it's going to feed back and tell the anterior pituitary to stop releasing LH and tell the hypothalamus to stop releasing gonadotropin releasing hormone. Follicle stimulating hormone is going to act indirectly to stimulate spermatogenesis. Together, FSH and testosterone act synergistically on those testaculaire or Sertoli cells, which stimulates the secretion of androgen binding protein, or ABP for short. That's released into the lumen of the seminiferous tubules and the interstitial fluid that surrounds the spermatogenic cells. Its job is to keep testosterone levels high, allowing for the final steps of spermatogenesis to occur in the seminiferous tubules. So that's how follicle stimulating hormone helps to stimulate spermatogenesis. The key is that it's indirectly doing it. It's not directly, but it's kind of helping get that process continued. We talked back in our endocrine lecture about inhibin, a hormone that's actually released by the sustentacular cells of the testes. It's going to inhibit follicle stimulating hormone by the anterior pituitary. So if, for example, we have plenty of FSH, we can release inhibin, and that's going to kind of slow that secretion process down. Testosterone, along with another hormone called dihydrotestosterone, or DHT, which is just a hormone made from testosterone conversion. Together, those produce several effects. Okay, they help with prenatal development, so development of the reproductive organs and whatnot that we just kind of talked about before the baby's even born. So development of the system ducts, the descent of the testes, development of external genitals, and possibly certain regions in the male brain. Testosterone and DHT also help with development of male sex characteristics like the enlargement of the male sex organs, obviously the penis being the key one, but also other organs as well. Development of masculine secondary sex characteristics like the enlargement of the larynx, the voice box, which explains why men typically have a deeper voice, along with things like the growth of a lot of hair on a man, especially when they hit puberty and they start growing facial hair, pubic hair, pretty much any and everywhere. Guys are hairy, and I can say that I'm a guy and I am very hairy myself. Development of sexual function, so things like sexual behavior, spermatogenesis, your sex drive or libido, and then the stimulation of anabolism, which we've talked a lot about that here lately, building up type processes like the synthesis of proteins, which results in heavier muscle and bone mass in men. Okay. Looking here at the next page, we could see this relationship traced back to the hypothalamus with that GnRH. Here's the anterior pituitary releasing the two hormones FSH and LH as a result of GnRH. Okay, FSH, you can see what it's going to do. It's going to promote the secretion or the formation of ABP. Okay, whereas LH is going to stimulate those interstitial cells to secrete testosterone. And we just talked about what testosterone and DHT do, which is what's shown down here in the purple box, which is essentially everything I just talked about. Over on the right side, they are showing you a feedback loop where blood levels of testosterone go up. And eventually what's going to happen is the release of less testosterone to bring those levels back down to normal, which is actually what's being shown here with the red lines that are coming back from the testes to the anterior pituitary and back to the hypothalamus. Not really anything that I'm going to directly pull from, but they were in your textbook, so I included them. All right, really quickly, we can talk now about the male sexual response, which is a 3-step process, and there's a lot of information here, and sorry, it's going to be kind of hard to read because there's so much, but bear with me. Starting with erection, this is a process, I'm sure everybody knows, where the penis becomes stiff and larger. Okay, happens upon sexual stimulation where the penis becomes enlarged and stiff, known as an erection. Okay, it is a parasympathetic reflex coordinated by the spinal cord. During an erection, parasympathetic nerves that extend from the sacral portion of the spinal cord to the penis release nitric oxide. Nitric oxide is that gaseous hormone that we've seen a few different times. It is a potent vasodilator. Okay. This causes the smooth muscle in the walls of the arterioles that supply the penis to relax, or in other words, to dilate, allowing larger amounts of blood to enter the blood sinuses of the erectile tissues. As the blood sinuses expand, the veins that drain the penis are compressed, which slows the flow of blood out. So essentially the blood flows into the penis, and then the blood kind of gets trapped. It's not going to be a forever trap, hopefully, but it will hold it in there for a period of time. Okay, and this combination of dilation of penile arterioles, expansion of blood sinuses, and compression of the penile veins results in the formation of the erection. Okay, this filling of blood in the chambers is called vasocongestion. Okay, this is also something that females experience in some of their reproductive structures. And major stimulus for erection is mechanically stimulating the penis. So essentially direct stimulation, touching the penis in some manner. Those mechanoreceptors that are found in the penis provide a direct input to the erection integrating center in the spinal cord. And we can see that right here on the next page, okay, where the penis has these receptors that can be stimulated through touch or whatever various means, triggers the sensory neurons that carry the signal into the sacrospinal cord, and as a result, output triggers the dilation of blood vessels. So more blood comes in, the penile veins be compressed, and the blood is going to stay in there. But keep in mind that the penis can become erect from other sources. Okay, and that's where we are looking at this portion of the figure, looking at erotic things, whether that's, you know, pictures or videos, smells, or even just thinking about sexual activity and whatnot can stimulate an erection. Okay, and so this involves descending inputs from the brain, things like the hypothalamus and the limbic system, okay, to the spinal cord. Okay, now negative stimuli like a bad mood, depression, anxiety can also inhibit an erection through these descending pathways. So that can affect sexual performance, obviously. The man can't get erect. Okay. Emission refers to the discharge of a small volume of semen prior to ejaculation. When sexual stimulation becomes intense, sympathetic nerves from the upper lumbar portion of the spinal cord stimulate contractions of the smooth muscles in the walls of the epididymis, the vas deferens, and the ejaculatory ducts. Okay, and this causes sperm to be moved from the testes, from the epididymis up into the vas deferens towards the urethra. Okay, same thing is going to be seen with fluids coming from things like the seminal vesicles, from the prostate, so on and so on. Okay, so that's what this next sentence is stating. In addition, sympathetic nerves stimulate accessory sex glands to release their secretions. Semen is formed as these secretions are mixed up with the sperm. And then the powerful release of the semen from the urethra to the exterior is what we call ejaculation. It's a sympathetic reflex. We'll literally just call it the ejaculation reflex. Okay. In this process, somatic nerves from the lower lumbar and upper sacral portion of the spinal cord stimulate contractions of what's called the bulbospongiosus muscle. This muscle is located at the base of the penis. It's what is shown right here. And it's going to be stimulated to contract repeatedly in a pulsatile manner. And that's why when semen is ejaculated, it's not just a steady stream. Like when you pee, it comes out and bursts. Okay. And it's because of this repeated contraction process of the bulbospongiosus muscle. As part of the ejaculation reflex, the smooth muscle sphincter at the base of the urinary bladder will close, which prevents any urine from being expelled during ejaculation. So in other words, if a man's ejaculating, there's no pee that's being released along with it. And that also prevents semen from going up and into the urinary bladder at the same time. Because the location of the ejaculation reflex centers in the lumbar portion of the spinal cord, it cannot be interrupted by inhibitory stimuli from the brain. So once that process starts, it is not possible to stop. And it's going to happen. So it's you know, like jumping off the high dive. Once you jump off the board, there's no going back. It's going to happen no matter what. Oops. All right, so that's it for at least right now with regards to the male reproductive system. We'll kind of talk more about some stuff at the end of the lecture, but we're going to transition now to talking about the female reproductive system, and we'll start the same way as we did with the men, talking about the primary female sex organs, which are the ovaries. Once again, we've talked about those ovaries, those organs before. And that's because of the fact that they are the main endocrine glands of the female system, secreting the female sex steroids, estrogens, and progesterone. They are also the site of gamete production in females, producing the female gametes. We would commonly call them eggs. They have a kind of fancier term called ova. Okay? And we'll get into that whole process here in just a second. And there are two kind of main regions to each ovary. The ovarian cortex, which is kind of the more important part with regards to what we'll be talking about here today. And then the ovarian medulla, which is just deep to the cortex, and it's made up of loosely arranged connective tissue containing blood vessels, lymphatic vessels, and nerves. Not a whole lot to talk about there. Cortex is a lot, or is important. There is a lot to talk about here. It is full of what we call ovarian follicles. So these fluid-filled bubbles that are where the eggs develop. Okay? Each ovarian follicle is going to consist of oocyte, which is an egg cell, in a various stage of development. And then the follicle themselves will be cells that surround the egg. And so you can kind of think of these like these big bubbles. And look, okay, let me just show you a picture here of an opened up ovary where we can see those follicles. And there's several different forms, and it's all dependent upon the stage of development: the primordial follicle, the primary follicle, the secondary, and the mature follicle. And inside you can see is a egg cell at some point in development. As you can see, as we go from primordial to mature, the number of cells that surrounds the egg becomes more and more, and as that develops and the follicle grows, it also starts to have this cavity called an antrum, where you find follicular fluid. And those 4 follicles are shown right here, and we're going to talk about those starting on this page, going back to where right after that slide we were just on. So the production of sperm was spermatogenesis. Here, the production of eggs is oogenesis. Now, we just talked about spermatogenesis about 10 minutes ago, so that's fresh on our minds. I will be honest, though, when I say that oogenesis is infinitely more complicated than spermatogenesis. Okay? There's a couple different reasons why. Oogenesis, defining it as the formation, development, and the maturation of an ovum. And then a big reason why this is so complicated is it begins before birth in females. So unlike spermatogenesis, which doesn't begin until puberty, oogenesis happens before a female is even born, or it starts to happen, I should say. It's not completed until well after birth, but before birth it begins. And just like what we saw with spermatogenesis, we're going to trace everything back to type of stem cell or germ cell here, what we call an oocyte, an immature epithelial germ cell that migrates to the ovaries during early fetal development. Once inside the ovaries, these germ cells differentiate into oogonia, which that's the plural form of the word oogonium. They are diploid stem cells, so they have 46 chromosomes, and they divide mitotically to produce millions of more germ cells. But most of these germ cells degenerate prior to birth in a process known as atresia. Okay? A few of those do develop into larger cells known as primary oocytes, which are the ones that begin the process of meiosis 1 by entering prophase 1. And this is still all, once again, during fetal development. Okay, so a primary oocyte is the first cell that's beginning meiosis. Okay, and if we kind of come here to the next page, we can trace it back once again to one of those stem cells called an oogonium. That's what divides through mitosis to form these primary oocytes. Notice they're both labeled with 2N at their center in the nucleus, because they still have at this point 46 chromosomes. Okay, this primary oocyte begins meiosis 1. It goes into the very first stage, and that stage once again is prophase 1. But notice it's not completed until after puberty. So there's this arrested stage of development where essentially everything gets paused from before birth until puberty. So, females probably go through puberty around, age 10, 12, 13, 14, somewhere in that neighborhood. Every girl's a little bit different. So it can be quite a long period of time where this arrested stage of development is going or happening. During this time, each primary oocyte is surrounded by just a single layer of flat follicular cells, and that structure is known as a primordial follicle, which we can see here in both pictures on the next page. Okay. At birth, approximately 200,000 to 2 million of those remain. But by the time that female reaches puberty, only about 40,000 are still present, and around 400 of those mature and ovulate during a woman's reproductive lifetime. Okay. And so there's roughly 400 mature released eggs that have the ability to be fertilized. That's quite a bit different than the millions and millions and millions of sperm that a male produces every day from the time that they go through puberty until the day they die. Okay. When that female runs out of her roughly 400 eggs, that's all she's got. She can't make more. And so at this point, they would technically be no longer fertile, no longer viable of having babies. Okay. And that is called menopause. So each month after puberty until menopause, hormones from the anterior pituitary stimulate the development of several primordial follicles, although only one of those will reach maturity for ovulation. A few of them will start to grow, developing into primary follicles, which consist of that primary oocyte surrounded by more layers of granulosa cells. We can see that here in letter B, as well as up here. So we still have our same egg cell on the inside, that primary oocyte. Okay. And we see several more layers of cells surrounding it. Okay. That primary follicle then will mature into a secondary follicle. Okay. And this is where we see the space known as the antrum start to form, where the cavity, that antrum, fills with the follicular fluid. Okay. Notice though, from figure B to figure C at the center, we still see what's labeled as a primary oocyte. Okay. The secondary follicle eventually becomes larger and forms into that mature or Graafian follicle, which is what is shown over here on the far right, the biggest one. Okay. Just before ovulation, that diploid primary oocyte completes meiosis one, which means we go from one cell to two cells, and each cell is going to be haploid. But what we see here in letter D with our secondary oocyte, which is one of those two cells, we don't see the other. And that other cell is called a first polar body. Okay, looking at the next page, you can really kind of appreciate what's actually taking place. This diploid primary oocyte forms these two very unequally sized cells. The first polar body is the small cell. The secondary oocyte is the important egg cell. This first polar body, just essentially consider it nothing. It isn't nothing, it's a cell, obviously, and you can see it does go through meiosis 2 and forms two more cells from it. But these cells don't really do anything. They just kind of die off. Okay. It's the secondary oocyte that's important. Okay. This cell has received most of the cytoplasm, which is why it's much bigger. And once formed, it begins meiosis 2, but then stops in metaphase 2. So it's a kind of weird situation again, where we start cell division, but we don't continue and finish it right away. This time it doesn't take 10 to 15 years or whatever it is from before birth until puberty. Okay. And I'll explain what and why this occurs here in just a second. Okay. Now, what's important about that mature follicle is that's what is going to burst open and allow the egg to be released into the fallopian tubes, into the uterine tubes, into the oviducts. All those terms mean the same thing, and these tubes that are connected to an ovary that lead to the uterus. This process where the egg is released into those fallopian oviducts, uterine tubes, whatever you want to call them, it's called ovulation. And every female experiences ovulation unless she's pregnant about midway through her reproductive cycle each month. We'll get into that here in a second. Okay. Now remember, that secondary oocyte, which is what's actually ovulated, has started metaphase 2, but is on pause, okay? So meiosis 2 in that oocyte only resumes when and if the head of the sperm cell enters the secondary oocyte. So only if the sperm has broken into the cell does it go through the rest of the stages of fertilization, okay? Which means now that secondary oocyte must split, which is what we see here and here. And we form an ovum and we form a second polar body. The second polar body is a lot like the first polar body in that we can basically just ignore it. Not really anything to worry about or important there. It's all just degenerated. Okay, it just dies off. The ovum is very important because this is the cell that has been fertilized by the egg. Okay. And will then, when the nucleus of the sperm and the nucleus of the egg, the ovum that is, unite, that's what forms into a diploid zygote, which is what we show down here at the bottom of our figure on the left. Once again, this is the cell that we all began our life as, one cell that then can divide, becoming 2 cells, that can become 4 cells, and so on and so on. And here we are, made up of trillions upon trillions of cells. Right. Now, after ovulation, going back into the ovaries, that bubble that was burst, that layer of cells that surrounded the egg, is actually going to continue to do something. That's going to form into a what we call corpus luteum. Okay? And it's going to produce those hormones that are very important to the female reproductive system and the cycle that I'll talk about in a second. That includes the hormones progesterone, estrogens, relaxin, and inhibin, and then it degenerates into a scar tissue known as the corpus albicans, which is what we see here. All right, hang on one second, guys. My wife is calling. I better answer it. Okay, so that's oogenesis. Okay, pretty complicated. Like I said, quite a bit more complicated than spermatogenesis. Looking at this figure, I kind of focused on this one on the left. Essentially, it's showing you the same thing over here on the right, which is kind of nice. I like this one on the right because it's breaking down what's happening just with the egg here in the little columns area for the oogenesis stuff, plus what's going on with the follicle that surrounds it, the bubble that's enclosing the egg. So it's the follicular development stuff, and might be handy to kind of help you study. Same thing as what I kind of told you for spermatogenesis. Make sure you know like which cells are diploid, which cells are haploid, know what like ovulation is, know what fertilization leads to, and all that wonderful stuff. So you're not gonna have to walk through all the steps of it, but make sure you know the kind of key points. Okay, so those were the primary female sex organs. Let's talk about the secondaries. I've already talked a lot about these first tubes or ducts, whatever you want to call them. Uterine tubes, fallopian tubes, oviducts. I dropped the word uterus a little bit ago. And then the vagina. The uterine tubes, fallopian tubes, oviducts, whatever you want to call them, are the active conduits for the ovum to reach the uterus. Their job is to transport the secondary oocyte away from the ovary once it's ovulated towards the uterus. Consists of 3 regions: an infundibulum, which is the very beginning, containing pulsating cilia, that help draw the secondary oocyte into the uterine tube after being expelled from the ovary. Ampulla is the widest and longest region. I highlight this, it's a very important thing, typically the site of fertilization by sperm. And then the isthmus is a narrow region that just leads to the uterus. Okay. Notice there is nothing about that egg cell staying after being fertilized, or so I should call it a zygote, staying in the uterine tubes and developing there, because that should be taking place in the uterus. Okay, the thick muscular chamber that harbors the fetus provides a source of nutrition for and expels it at the end of its development. This is why we call this commonly the womb, okay, where the baby develops and will hang out for 9 months until childbirth. Okay. There are times where the fertilized egg, that zygote, that forms from that fertilized egg, will develop in the uterine tubes. It sticks to the wall. This is called an ectopic or tubal pregnancy, and it is typically not a good thing at all. It's not a viable situation. A lot of times just leads to miscarriage and obviously death of the baby. Where that developing baby needs to be is in the uterus. And the wall of the uterus consists of 3 layers. The periometrium is on the outside. Physiologically, not much to discuss there. Myometrium is a very important layer made up of some muscle that helps with childbirth. That's where the contractions that a female experiences during the childbirth process comes from. It's actually the muscle pushing that baby down into the cervix, which is the narrow region at the bottom of the uterus that leads to the vagina. The inner lining of the uterus is called the endometrium. It's a highly vascularized mucosa lining the inside, as I've already mentioned. Consists of 2 layers, one of which is shed during menstruation. That actually, as of this newer textbook, they've kind of changed it, made it say 3 layers. But that's just because it kind of took one of the 2 layers and split it into 2 different sub layers. So it's got either 2 or 3 layers, but know that the menstruation process, when a female bleeds, it's because part of her endometrium has been stripped away. And we'll get into that process here in a little bit. Okay. Once again, the cervix is at the bottom of the uterus, leads to the vagina. You hear a lot about the cervix because this is what has to dilate and open up. And so when a female, they say she's dilated to like a 4, 6, or an 8 or whatever, they're talking about how open their cervix is. And so how much, that baby's gonna be able to fit through. It goes from about the same width as a pencil, as they say, to the width of the baby's head. So obviously very, very wide is what it can get to. And thankfully, after a period of time, it shrinks back to its normal size and position after childbirth. What's really cool, within the cervix, there's cervical mucus that's secreted that protects against bacteria that may enter the uterus from the vagina. And speaking of the vagina, it's a tube allowing for discharge of the menstrual fluid, receipt the penis and semen, and then the birth of the baby. There's 3 layers to its wall, adventitia on the outside, just kind of holding it in place. Middle muscularis, made up of 2 layers of smooth muscle, which permits expansion of the vagina during childbirth and sexual intercourse. And then an inner mucosa, where bacteria like to kind of chew up, and I say that very loosely. It's not actually literally chewing up, but bacteria eat up the glycogen that's stored in their mucosa cells, which ends up producing an acid solution that will lubricate and protect the vagina against microbial infections. And we've kind of talked about the acidic environment of the vagina back when we talked about the lymph and immune system. Okay, so I've kind of been alluding to talking about the female reproductive cycle. Let's do that now. During their reproductive years, so in other words, from puberty until menopause, non-pregnant females normally have or exhibit cyclical changes in their ovaries and their uterus, usually taking about a month, right around 28 days. Okay, and so we can actually kind of talk about things specific to the ovaries. We call that the ovarian cycle, a series of events in the ovaries occurring during and after maturation of the oocyte. And then what's going on in the uterus, which is sometimes and more commonly, I'd imagine, called the menstrual cycle, which is specifically talking about the changes to the endometrium, which is preparing it for the arrival of a fertilized ovum. Does it obviously receive a fertilized ovum every month? No, but it's a possibility, and getting ready for it if it does happen. Okay. So there's those 2 cycles, and then if you just kind of combine them, you would call that the female reproductive cycle. It encompasses both ovarian and uterine cycles, but also the hormonal changes that regulate them, and the related cyclical changes that occur to the breast and cervix. Once again, this lasts about 28 days and consists of 4 phases. We'll talk about those 4 phases and we'll get into the reproductive cycle in just a second, but let's learn about the hormones. Okay, as you can see, quite a bit of hormones. So not only is oogenesis much more complicated than spermatogenesis, but the hormones that regulate the female reproductive system and everything going on quite a bit more complicated than the hormones that regulate the male reproductive system. We can trace it back to the hypothalamus the same way that we could with the spermatogenesis process, and the hormone is the same one that we talked about earlier today, gonadotropin releasing hormone, GnRH, hormone from the hypothalamus that triggers and controls both the ovarian and uterine cycles. Its job is to strictly just stimulate the release of FSH and LH from the anterior pituitary. And yes, it's the same 2 hormones that men produce, although doing very different things. In females, follicle stimulating hormone stimulates the follicles, hence the name. Remember, the follicles are those bubble-like structures with the egg on the inside. Okay, so follicle stimulating hormone initiates follicular growth. So, like the cell division and the cell layers that form around the bubble or around the egg. Okay, that's what it helps stimulate, and it stimulates those ovarian follicles to secrete estrogens. Luteinizing hormone, the other hormone from the anterior pituitary, stimulates further development of the ovarian follicles, stimulates them to secrete estrogens, stimulates the follicles to produce androgens, and I would highlight this, triggers ovulation and the formation of the corpus luteum. So the kind of most important, not that the other things aren't important, but the really, really important thing here that luteinizing hormone does is it triggers ovulation. So popping the bubble, releasing that egg cell into the fallopian tubes, and in doing so, that bubble becomes the corpus luteum. Okay. Now notice it is spelled pluralized here, estrogens with an S at the end and not estrogen. People seem to use it singularly when it's actually multiple. There's a group of hormones that are classified as estrogens, secreted by those ovarian follicles and have several key functions. They promote development and the maintenance of the female reproductive structures and the secondary sex characteristics unique to women. So, like growth of the breast tissue, growth of pubic hair and stuff like that. I'm teaching to you ladies out there, so I don't have to explain what happens to your body when you hit puberty and become a woman. Estrogens also increase protein anabolism, including building strong bones. So that's a definite benefit. Unfortunately, that is why after menopause, it's more likely for women to experience osteoporosis, because they aren't releasing estrogens in high amounts, and so it doesn't help build their strong bones. Estrogens also actually lower blood cholesterol, which is a very important benefit, and if those levels of estrogens are elevated, it will inhibit the release of GnRH, LH, and FSH. Okay. Progesterone secreted by the cells of the corpus luteum. Once again, that's the structure that forms when that bubble bursts, when the egg is ovulated. Along with estrogens, it helps to prepare and maintain the endometrium for implantation. So in other words, for that growing fetus, it's not a fetus at this point, but it's a group of cells that will develop into the fetus to travel to the uterus and stick to its wall. Progesterone also is going to help stimulate the mammary glands to produce milk. And then it, just like with estrogens, its elevation or elevated levels will inhibit the secretion of GnRH and LH. Relaxin, we've talked about this before. Its name is pretty straightforward in explaining what it does. Released by the luteum during each monthly cycle. It's going to relax the uterus by inhibiting contractions of the myometrium, and at the end of pregnancy, increases flexibility of the pubic symphysis and dilates the uterine cervix. So it gets the body ready to go through childbirth. And then inhibin sounds like the word inhibit. Just like the hormone in men, it's released this time by granulosa cells of the follicles, as well as the corpus luteum after ovulation, and will inhibit the secretion of FSH and to a lesser extent, LH. And I'll give you a figure here showing you some of those hormones and how they cycle and go up and down with regards to what's taking place here with the 4 phases of the reproductive cycle. We'll get to that figure in a little bit. So now we can talk about the 4 phases. It'll span us a couple slides here to get through it all. All right, the first phase of the 4 phases is the menstrual phase. It's days 1 through 5 approximately. And I've broken things down into what's kind of going on in the ovaries and what's going on in the uterus. Okay. In the ovaries, under the influence of follicle stimulating hormone, several of those primordial follicles are developing into primary follicles and then into secondary follicles. Now, this process is not able to be completed in 5 days. It takes several months, which means that a follicle that begins its development in one cycle will not mature for several cycles later. Okay. Now, this is the phase that's really more related to what's going on in the uterus. And it's called the menstrual phase because this is when the female's going through her menstrual discharge, what people commonly call a female period. Okay. It's because of declining levels of estrogens and progesterone, which stimulates the release of prostaglandins that cause uterine spiral arterioles to constrict. So it's basically what's happening is the blood vessels in the endometrium are pinching shut. And so the cells become deprived of oxygen, leading to their death. So at this point, part of the endometrium is being lost, and that's where the bleeding comes from. And upon completion of this phase, only the stratum basalis, which is one of those layers of the endometrium, is what remains. Following that, the next week or so, days 6 through 13, we have what we call the pre-ovulatory phase. We call it the pre-ovulatory phase because it's before ovulation. So it is the time period between the end of menstruation and the beginning of ovulation. And the ovaries, some of those secondary follicles begin to secrete estrogens and inhibin. By about day 6, a secondary follicle in one of those ovaries has outgrown all the others and has become the dominant follicle. Estrogens and inhibin secreted by that dominant follicle decrease FSH, causing other follicles to stop growing. That is, unless multiple secondary follicles become codominant, which means that more than one egg is ovulated, which means that more than one baby can be formed. This would result in fraternal or non-identical twins. Something that's pretty rare. My oldest child, he's in a class with triplets. There's a boy and 2 girls, but you don't see that very often. So it's kind of unique. And we'll talk a little bit more about fraternal and identical twins at the end of the lecture, but not a typical thing in humans. Now, if you think about other animals like dogs and cats, they have litters, right? So their females are releasing multiple eggs at a time. And that's why, you know, sometimes you have like 6, 8 babies that are born. Normally, one dominant follicle becomes that mature graphic follicle, which means only one egg is ovulated. Okay. In the ovarian cycle, both the menstrual and pre-ovulatory phases are called the follicular phase because this is where the follicles are growing. In the uterus, estrogens released by those growing ovarian follicles stimulate the repair of the endometrium. Remember back in the menstrual phase, the endometrium lost part of its cell layers. It lost the stratum functionalis. All that was left is the stratum basalis. And so during this phase right after that, it's starting to build back up. The stratum basalis, that layer that was left, undergoes mitosis, and that forms a new stratum functionalis. And by the end of this week or so, the endometrium has doubled its thickness. Okay. In the uterine cycle, the preovulatory phase is called the proliferative phase because the endometrium is proliferating, it's growing. Day 14 of the 28-day cycle is when the female is going to ovulate her egg. Ovulation is defined as the rupture of a mature Graafian follicle, and therefore the release of the secondary oocyte into the fallopian tubes, caused by high levels of estrogens exerting a positive feedback effect on cells that secrete LH and GnRH. Remember I told you, as we talked about luteinizing hormone, it is the hormone that most directly triggers ovulation. Well, high levels of estrogens trigger high levels of LH, and you'll see that here. Actually, just show you on the next page. Looking here on the next page, we see LH and FSH peaking around day 14, which is when that female pops that bubble and the egg gets released. Okay. Obviously, ovulation is strictly kind of related to what's going on in the ovaries, so I'm not even going to really mention anything in the uterus. It's only a day anyway. But following that, the last 14 or 2 weeks, 14 days or 2 weeks, days 15 to 28, we have the post-ovulatory phase. So pre means before, and as we know, pre-ovulatory came before ovulation, post comes after. And the ovary, after ovulation, the mature follicle collapses, forming a corpus luteum under the influence of LH, which is going to secrete hormones, progesterone, estrogens, relaxin, and inhibin. So that bubble that pops becomes the luteum and will continue to release hormones. And for this reason, we call this part of the ovarian cycle the luteal phase because it involves the luteum. Okay. Now, what happens next is dependent upon if the female has gotten fertilized. If the egg is not fertilized, the corpus luteum lasts about 2 weeks before degenerating into the corpus albicans. Corpus albicans is essentially just a non-functioning scar that's left over that will eventually just kind of die away. And as a result, that corpus albicans is not going to be releasing hormones. So as the levels of those progesterone, estrogens, inhibin decrease, release of GnRH, FSH, and LH rise because there's a loss of negative feedback. Follicular growth resumes as the new ovarian cycle begins. If that oocyte is fertilized, though, corpus luteum lasts more than 2 weeks, and that's because of a new hormone that's released called human chorionic gonadotropin, or HCG. This is produced by the chorion of the embryo, which is a layer that embryo, that developing baby, we can call it a baby at this point, it's probably not a good accurate description of it, but the growing layer of cells or growing ball of cells takes place about 8 days after fertilization has occurred. This is the hormone that when a female pees on a stick, they're testing for. Okay, so if you've ever seen a pregnancy test or the box that a pregnancy test comes in, more than likely you'll see testing for HCG, human chorionic gonadotropin. Only if a female has a growing embryo should they be releasing it, which is why it's an easy way to detect if a female's pregnant. Okay. So that's all going on in the ovary and the uterus. Progesterone and estrogens produced by the luteum promote further growth of the endometrium. Because of the secretory activity of the endometrial glands, we call this the secretory phase with regards to the uterine cycle. Changes peak about 1 week after ovulation when a fertilized ovum might arrive in the uterus. But if fertilization does not occur, the levels of progesterone and estrogens decline because of the degeneration of the luteum. And I would highlight this last thing: withdrawal of estrogens and progesterone causes menstruation to start back over. And that's what we can see here on the next page. What I've essentially done is kind of combined two figures. It's not perfectly lined up, but figure A, so from here upwards, we're showing you what's happening throughout the 28 days, plus we started over and show the beginning of the first 2 days of the next round. Here we have this specifically showing you the uterus or menstrual cycle things, and then with regards to what's going on in the ovaries. And what I've done is I've tried to kind of stretch this graph to match up with the same days, and this is showing you what's happening with the hormones. Okay, we are not showing every hormone, but we see FSH, estrogens, LH, and progesterone. Okay, as you can see, during the first 5 days, all the hormones are pretty low in number. And once again, the first 5 days are really all about what's happening in the uterus with the loss of the layer known as the stratum functionalis. That's showing you here in our picture the 2 layers, which I said is now being described as 3. But this thick layer here that you see at the end of the cycle is what's lost during the beginning of the cycle. Those first 5 days when a female is on her period, it's because of the loss of these cell layers, this cell layer. Okay. What's taking place in the ovaries is just the development of those primordial follicles, primary follicles, da 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 da. As those cells or those bubbles develop, they start to release more and more estrogens, and as we can see, their levels go up. Okay. Following the 5 days of menstruation, the female really starts to just kind of replace that layer of cells that was lost. And like what we say about double the thickness of the endometrium occurs during that 7 days of post or pre-ovulatory phase. Okay. Now, day 14 roughly is when the follicle bursts. That ruptured follicle releases the egg, which we already pointed to because of high levels of estrogens and LH, luteinizing hormone, which results in the egg being released into the fallopian tubes, but also the formation of that corpus luteum, which is going to, as you can see, release hormones like progesterone and estrogens, and that's why we see progesterone really rise up, and then not quite as high as it was back here, but we still see elevated levels of estrogens. Now, once again, this will last about 2 weeks before turning into the corpus albicans, which basically is dead group of cells, nothing really going on there. That is only if a female's not been impregnated. Okay, if they have been impregnated, this will continue to release those hormones. But when that corpus luteum becomes a corpus albicans, if the female does not get pregnant, the levels of estrogens and progesterone go back down, which is what triggers ovulation or menstruation to begin again. So I'm sure you've heard in like movies and TV, hopefully nobody's ever told you this in your life, but you know, the phrase that females can be very hormonal. I think that's a very bad way to try to put down a lady. Okay? Obviously, as you can see now, females are very hormonal, but in a very amazing way. Okay? Making babies, producing the eggs to produce a baby, developing the uterus, and going through all those stages every month during those reproductive years. It's a very amazing thing. And I'm obviously not a female, but I can tip my cap to you guys because, you know, I find it very amazing. So if you ever are told that you're being hormonal, I'd say thank you, because it's amazing. Now, if you really want to confuse the heck out of yourself, try to work yourself through this figure where it's showing you all the different feedback loops with all the different hormones, all the different structures, the follicles there in the middle of the uterus, down there at the bottom. We see not only red lines there to represent negative feedback, we also see the green lines representing positive feedback. I'm not even going to work through it just because I don't want to confuse you anymore. I'm sure after running through all those 4 phases, I've confused you enough. I really only included it because it was in your textbook and as another way to just demonstrate how complicated but amazing the reproductive cycle in a female really is. So with that said, we're going to transition away from talking about the female reproductive system, kind of talking now about making babies. Okay. So let's talk about ways that we can affect fertility. If you don't know what fertility is, fertility you can think of as just the possibility of fertilization, which those words sound very similar. They're basically the same word in just different form. Fertilize, fertilate, fertilization. not fertility, that's not a word, but fertility, that's a word. Okay. So when I say factors affecting male fertility, I mean factors that are going to affect a male's ability to produce viable sperm and therefore make possible fertilization in a female. Okay. One of the most important factors affecting males' fertility is the number of sperm that they produce. A low count of sperm and or a high proportion of abnormal sperm is a major cause of sterility, which is where a male is considered to be not fertile, which seems kind of weird because somebody that's sterile is still producing millions upon millions upon millions of sperm, but it is much less than what you see in a normal sperm count. Okay, per milliliter, remember men are going to ejaculate about 2.5 to 5 mils of sperm each time. Each of those mils should have about 100 million sperm. Okay, but if they have less than 20 million, they are considered to be sterile. Now, does that mean that they have no chance of making a baby? Obviously not. They're still producing 20 million sperm per milliliter, but it's a much less number than normal and a much less likelihood that they would be able to fertilize a female. Now, a thing that can affect their sperm number is how often a day the man ejaculates, okay? Which should make sense. The more times a day a man ejaculates, the fewer and fewer sperm would be in those mils. So in other words, if a man and a woman were trying to get pregnant, they were having sex multiple times a day, really the first couple times is going to be the highest possibility of it because of the fact that it affects the sperm number. Now, we talked the other day about the importance of the scrotum to keep the testes in an environment, somewhere a little bit cooler than core body temperature. For optimal sperm formation, it's need roughly to be around 32 degrees Celsius, which is roughly 5 degrees below core temperature. If the testes are kept inside the body or too close to it, it's going to result in the formation of sperm ceasing. That can happen like when a man wears tight clothing, like tighty whities, as they say, reduce the sperm count in men. Spending just like 30 minutes in a hot bath or like in a hot tub can lead to a 90% decline in sperm number. But obviously, if they get out, and cool off, then they're going to rate that production sperm back up. Additional factors that can affect male fertility include the excessive consumption of alcohol, major stress, being malnourished, and certain drugs, which most cases are just going to reduce fertility by reducing sperm count. Radiation decreases male fertility, and I don't know why. I've never really done the research, but sperm production is typically higher in winter, regardless of scrotum temperature. And I always joke, I guess if it's cold outside and there's snow on the ground, there's no other alternative but to just stay inside and make babies. So it's a good time of year to do it. All right, there are obviously factors that affect the likelihood that a female can get pregnant. So let's talk about factors affecting female fertility. All right, the first factor is not only the age of the egg, but also the age of the sperm that have been released inside of her. Okay. Sperm in the female reproductive tract undergo aging but remain motile and do have the ability to fertilize for up to 4 days, which explains why a female doesn't necessarily have to be on day 14 in her cycle during ovulation for her to get pregnant. And now freshly ovulating eggs are mature and best for fertilization for about 12 hours after ovulation. They do age gradually and become less likely to be fertilized after that, but it is not unheard of for a female to get pregnant when having sex at pretty much any point during the reproductive cycle. And just best, and that's why, if a female and a male are trying really hard to get pregnant, making sure they have sex on ovulation day is very important. Okay. A female's age affects their fertility. It's highest in their 20s and early 30s, declines beyond that in the late 30s to very low levels in the 40s. And then somewhere around age 50 is when a female ceases fertility and therefore has reached what we call menopause. And at this point, they are no longer fertile because they aren't producing and releasing any eggs. Menopause is characterized by a cessation of menstrual cycles, ovulation, fertility, and pregnancy. Hormone intervention is something that can affect fertility. Injections of gonadotropins, LH, and FSH or ACG would enhance the number of follicles developing in the ovaries, as well as the chances of ovulation. Therefore, in this case, a lot of times people that do go through these hormonal interventions are more likely to have multiple births, not, like get pregnant multiple times, but have, multiple babies, so twins, triplets, that kind of stuff. Very common for females to utilize contraceptive devices and contraception. As far as contraceptives go, they aim to reduce fertility or the chance of pregnancy in a variety of ways by preventing ovulation. That's what, for example, the pill does, that a lot of females utilize. Other contraceptives are going to reduce and prevent the encounter of sperm and egg. Also things like fertilization or implantation. I'm sure you've heard of like a diaphragm that can be utilized to basically block the sperm from fertilizing the egg because it's the place where the cervix and the vagina meet. I'm sure you've heard of like Plan B or like the day after pill. That's something that would affect implantation. And there's other things as well. And obviously females aren't the only sex that can do contraceptives. Men can wear condoms and do different things, but contraceptives are probably a lot more common, I would say, in females, including the pill. Oral contraceptives, like the pill, containing synthetic estrogen and progesterone, inhibit the release of follicle-stimulating hormone and luteinizing hormone, which thus prevents follicular growth and ovulation. So that's the factors that can affect fertility. Let's talk a little bit more about what fertilization entails. Fertilization, we've already talked about this before today and the other day as well. Defined as the penetration of a secondary oocyte by a sperm cell, resulting in meiotic division of that secondary oocyte, forming an ovum, and then the subsequent union of the nuclei of those two gametes, resulting in the formation of a single diploid nucleus inside of that zygote. And that zygote with its diploid nucleus has how many chromosomes? 23 pairs for a grand total number of 46. Good. This generally occurs in the uterine or fallopian tube within 12 to 24 hours after ovulation. Sperm that reach the vicinity of the oocyte within minutes after ejaculation are actually not capable of fertilizing it for about 7 hours. And this is because they must undergo that process of capacitation, which I mentioned earlier, is a series of functional changes causing a sperm's tail to beat even more vigorously and prepare its plasma membrane to fuse with the oocyte's plasma membrane. For fertilization to occur, a sperm cell must penetrate 2 layers. And these are 2 layers on the outside of the egg. One is called the corona radiata, which are granulosa cells surrounding the egg, and then a zona pellucida, a clear glycoprotein layer between the radiata and the oocyte's plasma membrane. Now, remember, on the head of the sperm is that cap-like vesicle structure called acrosome, which releases these enzymes that help to penetrate the radiata and come into contact with the pellucida. Okay. On the zona pellucida are special receptors called sperm receptor ZP3, which I don't have any idea why they call them that, but they are a receptor for sperm. I guess that makes sense. I don't know where the ZP3 stuff comes from. But once bound, that egg's plasma membrane will just like suck that egg or suck that sperm in. And this is the main stage of fertilization. A reaction called the zona reaction occurs where there's a rapid chemical modification of pellucida, which will block the penetration by any more sperm. So it will prevent two sperm from fertilizing that same egg. That's not what would produce twins or anything like that. Now, once the sperm cell enters the secondary oocyte, the oocyte must then first complete meiosis II, because remember, it's stuck in metaphase 2. And so that secondary oocyte must become the ovum. Okay. The nucleus in the male's sperm, or in the head of that sperm from the male, becomes the male pronucleus. The nucleus of the fertilized ovum develops into a female pronucleus. At this point, the two nuclei are separated, and so the chromosomes are still separated, but then those nuclei fuse. So they're both called a pronucleus, so they fuse together, and we call them pronuclei fusion, and that results in the recombination of the chromosomes. And in other words, the 23 from the egg, 23 from the sperm come together, forming that diploid zygote. Now, there's a fancier term for both fraternal and identical twins, and we would use the terms dizygotic and monozygotic to describe it. And it describes how many zygotes are involved. In the case of fraternal twins, dizygotic means 2 zygotes, because di means 2, dizygotic. So in the case of fraternal twins, it's 2 different secondary oocytes fertilized by 2 separate sperm. And that explains why dizygotic twins can look very different. Not only that, but they can also be the complete opposite sex. Whereas monozygotic, what does mono mean? 1. And so in a monozygotic or identical twin situation, it's 1 single fertilized ovum. And therefore, what happens is during the development of the embryo, from that 1 sperm, 1 egg, it splits. And therefore, you end up with 2 identical twins. They have to be the exact same gender. They're going to look exactly alike. And that's because they have the exact same DNA. Okay. The key here is that when they develop, they separate into 2 different embryos. Does that make sense? Now, you can have identical triplets. That's super rare, but it does happen. So it'd be 3 embryos that split and formed from that original single ovum. Okay. I don't know if it's actually possible, but in theory, I guess it would be if you had 2 fraternal twins, and then one of those twins, that embryo split. So you could have triplets with 2 of the kids being identical twins, but I'm guessing that's really, really rare. Okay. That'd be pretty crazy. All right, what comes next? Again, all that stuff still taking place out in the fallopian tubes. After fertilization, and I'm talking about 24 hours later, roughly, so about a day later, rapid mitotic cell divisions of that zygote called cleavage take place, where we go from 1 cell to 2 cells, and by the end of the 3rd day, we've got about 16 cells. And that leads to the formation of what's called a morula, which is about day 4. And then about day 5, this solid ball of cells known as a morula becomes a blastocyst, which if you look internally, you see that it's a hollow structure, a hollow ball of cells, which is different than a morula, which is a solid ball of cells. But this blastocyst, I keep looking at the wrong word and saying it. This blastocyst is important because it's what undergoes implantation, which is where it's made its way into the uterus and sticks to the wall. Okay. This takes place about 6 days after fertilization. So not quite a full week after fertilization, but this blastocyst is going to stick to the wall of the uterus, and that's where it's going to remain for the remainder of the intrauterine life, for the remainder of the life inside of mom. Okay. Now, at this point, the blastocyst consists of 2 parts: the inner cell mass, which will become the fetus, the surrounding membrane known as the chorion, which becomes part of the placenta. Remember, the chorion is what released that HCG, human chorionic gonadotropin hormone, that we mentioned earlier. Okay. And it becomes part of the placenta, which is a special structure through which the exchange of nutrients and waste between fetal and maternal circulations occur. It's also known as afterbirth because this is what actually has to be pushed out of mom after she's given birth to the baby. In humans, it's kind of used for like testing purposes, but in like other animals, it's pretty common for mom to eat it. Like in cows, that's very common. Sounds really gross, but there's a lot of nutritional benefits to mom from it, and that's why it's not just wasted. The umbilical cord should not be confused as part of the placenta. The umbilical cord is the actual connection between that placenta and the fetus. So the placenta grows in the womb, inside the uterus, with inside the uterus, just like the baby does. And then connecting the placenta to the baby is the umbilical cord, which when the baby's born, they have to cut. And that's what leaves the scar known as the umbilicus navel, or what most people would call the belly button. Okay. Now, about 15 days after fertilization, a process called gastrulation occurs. We call it gastrulation is because it forms from a blastocyst into a gastrula. And this is where we first see the three primary germ layers develop. This is the first time we'll see any tissues. And those three tissues are the ecto, the meso, and the endoderm. And if you look at the next page here, table 29.1, it lists off the structures that develop from these three original germ layers. Okay. For those of you who are anatomy, if you remember the beginning of the semester, I drew this saying that looked like a Pac-Man with three cell layers. Those three layers are the endo, the meso, and the ectoderm. But from that inner layer of cells, the endoderm, come all the structures that are listed here. From the middle layer of cells come all the structures listed there, and then from the outer layer of cells, all the structures listed there. I do actually want you to study this table, and I'm sorry that it's kind of this way where it's got the colors, because I know when I printed it for you guys, it's kind of black and white, a little harder to tell. So you might want to maybe pull this up digitally where you can kind of read it a little bit better and study it that way. Now, what takes place next from the 4th through the 8th weeks of development are very significant when it comes to embryonic development. This is called organogenesis because this is when the organs are developing, appearing, and it's all super early in fetal life. You know, 4th week, 8th week, and you know that's not even through the first trimester before we've seen the organ systems coming in. By the end of that 8th week, all the major body systems have begun development. All functions may still be pretty minimal. By the middle of the 4th week, the upper limb buds begin to develop. Lower limb buds and the heart prominence form. At the end of the 4th week, the tail develops on the embryo. During the 5th week, the brain and head develop rapidly, the limbs develop further. By the 7th week, the regions of the limbs become distinct and digits appear. By the end of the 8th week, eyelids come together, the tail disappears, external genitals begin to differentiate, and digits are distinct and no longer webbed. Do you need to know every single thing here? No. And if you want to learn more about this, I know some of you guys probably have already taken this class, but Human Growth and Development is a great, wonderful resource and a great class to learn a lot of this stuff. Just make sure you know what organogenesis is. Once again, the development of the organs and organ systems. And then here, it's our last slide, right? Yeah, the last slide, briefly introducing the fetal period. Once again, if you take Human Growth, you'd learn a lot more about this. But this represents the time from the 9th week until birth. And during this time, tissues and organs that develop during the embryonic period grow and differentiate. But we see very few new structures appearing. It's just really body growth and development take place, not so much new structures, especially in the last half of intrauterine life. Okay, so in the last 4.5 months, not a lot changes other than just like size and growth and whatnot. For a more specific breakdown on what's taking place, you got this nice, wonderful table here, breaking it down for the 9th through the 12th weeks, 13th through the 16th, all eventually down to the 35th through 38th weeks, and typical changes that have or are going to occur. But nothing I'm going to pull from on the table that you need to know. Just know what the fetal period is, kind of the main thing I'd like for you to know. Like I said, take human growth and development, you'll learn a lot of good stuff. But that's not what this class is, so not going to worry about it.